Have you ever wondered how we make sure medicines remain safe and effective throughout their entire shelf life? Well, that's where forced degradation studies come in. In today's video, we'll explore what the ICH Q1A R2 guideline expects and why these studies are so important for pharmaceutical development. Let's start with the basics. Forced degradation studies aren't just another regulatory checkbox. They're like a stress test for medicines, pushing a drug to its limits to see how stable it really is. At Resolve Mass Laboratories, we carry out these studies with precision, ensuring that every data point not only meets global standards, but also helps scientists build more stable and reliable formulations. So, what exactly happens in a forced degradation study? Imagine taking a drug and exposing it to extreme conditions: strong acids, high heat, intense light, even oxidative environments. The idea is to make the drug break down on purpose. By doing this, we can uncover how it degrades, what byproducts are formed, and whether our analytical methods are good enough to catch even the smallest changes. Now, you might ask, why do we need this? Well, there are several reasons. First, it helps us identify degradation products, which is crucial for impurity profiling. Second, it allows us to design analytical methods that are stability indicating, meaning they can reliably pick up on changes in the drug. Third, it helps scientists understand the drug's weak points so they can improve formulation design. And finally, it supports regulatory compliance and shelf life predictions. In short, these studies are the foundation of a drug's long-term safety. Now let's talk about the rule book. According to ICH Q1A R2, a proper forced degradation study must cover different stress conditions: acid, base, oxidation, heat, light, and humidity. But here's the catch. The goal isn't to completely destroy the drug. Instead, the study should aim for 5 to 20% degradation. That sweet spot gives us just enough breakdown to learn something useful without making the sample unusable. And of course, every detail, pH, temperature, solvents, and exposure time, must be carefully recorded. Each stress condition tells a different story about the drug. Acid hydrolysis shows us how it reacts in acidic environments. Base hydrolysis checks its tolerance to alkaline conditions. Oxidation reveals whether it's prone to oxidative damage. Thermal stress answers the question, will it survive high temperatures? Photolysis shows us how sensitive it is to light exposure. And humidity stress helps us understand how moisture impacts the product. Together, these conditions paint a complete picture of the drug's stability profile. Now, why does this matter for regulators like the FDA or EMA? Because these studies prove that the drug won't lose potency or form harmful byproducts during its shelf life. They also show that the manufacturer is applying quality by design principles, which is a big plus for faster approvals and global acceptance. So what's the best way to approach a forced degradation study? Start early in the development process, ideally during method development. Aim for that 5 to 20% degradation window for meaningful results. Use more than one analytical technique because relying on a single test can miss important details. And above all, keep detailed, transparent records of everything. These practices not only meet ICH standards, but also save time and reduce risks in the long run. At the end of the day, forced degradation studies are more than just a scientific requirement. They are a powerful tool for ensuring that the medicines we rely on remain safe, stable, and effective throughout their entire life cycle. By following ICH Q1A R2 guidelines, companies can strengthen product reliability, reduce regulatory hurdles, and ultimately deliver better healthcare solutions. At Resolve Mass Laboratories, we specialise in these studies, along with HPLC analysis and analytical method development. So, if you're looking for trusted expertise in forced degradation testing, reach out to us today. Because when it comes to drug safety and stability, precision matters.